Hey, welcome guys. Welcome to Unknown. Today we will be talking about, or we will be experiencing about how to identify a bacteria, how to identify a bacterial species. So, bacterial identification, which is a basic requirement for any microbiologist to work in various aspects of microbiology. So, we will see the flow through, or you can follow this flow through for most of the bacteria. As I have shown here, all bacterial identification will start from mother culture and purification of the culture, that is, obtaining a single species through quarantine streaking, then having a first glycerol stock, meaning when you are subjecting any bacterial species for characterization as a molecular analysis, better have a backup first and then start working. Check your glycerol stock is, whether they are really working or not by reviving next day. Then make two more glycerol stock. It will be ideal to have three glycerol stock per species of a bacteria that you are subjecting to identification. Always document the results. The results that you are observing for each and every assay should be documented. It starts from basic gram staining, then various biochemical assay. From the biochemical assay and by comparing with the Burgess manual of dermatology bacteriology, we will be identifying the bacterial species. I will be explaining each and every step, how to document, what to do, what not to do, basically various assay involved in it. So in any characterization assay for bacteria, you have to follow three different stages. I would say this requires, multiple skill sets. You need to know how to handle the microscope, how to study the bacterial morphology. Same way, you need to know the chemistry behind each and every biochemical characterization you are doing, and then you should be having, molecular biology skills to isolate DNA or any whatever is essential for that study. So here I basically divide them into three categories: macro and micro morphological analysis, chemotaxonomy. I would say these two assays usually will give bacterial family or genera, but for species diagnosis, you need to go for molecular taxonomy, which involves sequencing 16S rRNA gene. This is the molecular marker for all bacterial species. So let me start from the beginning, starting with um, mother plate. Imagine you are isolating bacteria from a soil, water, plant leaf, whichever source it may be. The first resultant plate with culturable microbacterial community, especially, are called as mother plates. From there, you will study the morphology of each and every unique bacterial isolate in the mother plate. You can sample them and then go for foreign sticking. Here, what I have displayed is a series, bacterial mother plates obtained from serial dilution. So you you can, when you plate bacteria by pore plating, same sample by spread plating, you will have different bacteria. That's what I'm trying to say. So usually when I start bacterial identification, I will have a clear cut work plan. What are the assays I'm going to do, when I'm going to accomplish them. Have a defined work plan. This is called visualization technique. Visualize the entire workflow that you are going to do for identification. Then you enter the lab. It is very much essential to document simultaneously when you are observing each and every result. I'll be discussing on documentation. So once you got the mother plate, say here we have a mother plate, right? So identify the uniqueness of various isolates. Give the number 1, 2, 3, 4 based on their unique morphology. Each and every numbered unique bacteria go for foreign sticking. Foreign sticking is nothing but, you make four different quarants. You make a motherland here, and from here, I would say there are various ways to do it, but this way, I would recommend that you will get an isolated colony, which is an ultimate objective of doing a forensic. The last quadrant matrix is a way to see whether your bacteria is unique or not. So better get an isolated colony so that you can study the colony morphology. So I would start from motherland. I will make a thick bacterial smear. You incinerate the loop after that, cool it down. From here, cool it down. It's a simple word, but many times I see discontinuous growth because people are using hot glue. So better touch the uninoculated area in the agar, transfer the excess heat if present in your nichrome loop, then go for foreign sticking. I would suggest go for 6 lines from the motherland, incinerate, cool it down. From the last 3 lines, make more 6 more lines in the second quadrant. See, from the last three, four lines, you can make another line, and then if you do this, I would guarantee 99.9% of time you will get a isolated colony in the last quadrant. So that's how quadrant sticking is done. These are the things you can do. These are the things you should not do. Okay. While what has happened here is, the loop is hot. Very simple explanation. The loop is very hot. That's why there is no growth in the second quadrant. Here, I would say the candidate did not have a consistent strategy. Here he has used from last three lines, but here he has lost his focus. In the making of fourth quadrant, he has touched the motherland. That's why there is no isolated growth. He was supposed to get isolated here, but because he has somehow touched the mother lawn, again the continuous growth as a result. Do not document something like this, putting in the chair or something, take a picture. No. So this is how you are supposed to get an isolated colony, but there are some bacteria, for example, Bacillus megaterium. This is a quadrant sticking. The candidate did not do any mistakes here. But what has happened is Bacillus, megatherium, serious, sometimes they have a swarming motility in the plate in search of nutrients. You cannot stop them. They will just produce excessive XFL or polysaccharides, and they will fill the plate. Very much hard to get the isolated quality. But in my experience, when I increase the agar concentration to 1.5% to 3 or 4% agar, you will control the swarming activity and you will get isolated quality. So it is very much essential to have a very sharp skills with quadrant sticking, because this is the first basic quality for a microbiologist, that is to obtain isolated colony in the last quadrant. Once your skill is very good, you can play with the agar. I'm very proud to say one of my students, Nanda, he has made this picture. Louis Pasteur, this is using a streptomyces species in actinomyces isolation agar, and he has, this is his signature, Nanda Kumar. I'm really proud of this picture. And here is an agar art competition conducted by the American Society of Microbiology every year, where they will be conducting such competition. This is the pictures you are seeing made out of bacterial growth, growth chromogenic and non-chromogenic bacteria. They have used to make the picture. So the basic point is you need to have a good inoculation loop skills, so that rest of your experiments will be of good quality. And regarding wrapping, there is always been a debate whether to wrap the plates or not to wrap the plates. I have conducted very basic research to see the effect of the quality of wrapping on the growth of various bacteria. I will share that link along with this video. You can check it out. There is no much difference. Okay, especially in academic settings, if you wrap a place, first thing, your plates will be safe, free from Drosophila melanogaster, first of all. Even though some people or many people who is incubating their plates along with your plates, if they are not wrapping, they will attract Drosophila. But this will also put your plate in the risk. So better wrap the plates. Second thing, especially in academic setup, if you do not wrap the plate, especially you are using Brussels plates, when you open the incubator, it is high chances are that plates can, when especially the incubator is full, they can just slide off the incubator and get broken. So if you are inoculating or if you are introducing the wrap around the rim of the plate, this will cause friction and this will prevent sliding. So the first step in bacterial identification is getting a isolated colony. Wrapping, not wrapping, what kind of material you are using, all this will facilitate in getting an isolated colony in the last quadrant metric. Once you got the colony, the first thing you do is study its morphology. How it is it circular, regular, rhizoid shape? How is the margin of the colony, low base or filiform? You need to study, make a note of it in your observation, write it down. Whatever colony is assigned off or you are targeting to identify, you write it down. Then once it is written and it is documented, take a picture of the front plate or reverse plate. If you are studying morphology, front plate is mostly recommended. Front plate picture, you take it out. Once the picture, everything is done. Now remove the seal, open the plate for gram staining. The ultimate objective of gram staining is to see whether it's bacteria. First of all, to study the morphology, whether it's the cocci or coccobacilli or bacilli. Then we need to know the cell wall composition, whether they are rich in polysaccharides or lipopolysaccharides. Okay, they are rich in peptidoglycans. The gram positives are rich in peptidoglycans, which make them, purplish in color. Usually, in my experience, I see whether the bacteria is correlative or non-correlative spore-forming bacteria. You can determine easily during gram stain. Example, see here. You can clearly see vacuum space, within a cell. Only the polar region of the cell has been stained. Usually, this is an indication of endospore formation. Even here, it is visible. Okay, so those bacteria here it is not visible. So I don't know. So once you subject these bacterial stains, you will see the endospore. The ultimate objective of gram staining is to get isolated single cell. Okay, you should be able to observe somewhere in the field of view. You should be able to see a single cell. Then you can evaluate whether they are gram positive or not. Purplish, gram positive. This is an ideal, gram positives. Mostly they are gram positive. Yes. All right. What not to do? See, nothing can be derived out of such an observation in the microscope. Especially, even though their cells are good, you can see a slightly or dull, different contrast around the cell structure. This can be reduced if you can bring the condenser lens position very much closer to your slide. Many people they are adjusting both coarse and fine adjustment in the bright field microscope. What they are forgetting is to bring the condenser lens or adjusting the diaphragm, that is the intensity of light. These things can be manipulated to get an ideal structure. You see individual cells here. They are very clearly gram positive cells. This is the observation you need to make. Again, I am very proud of one more student who shot this. Actually, this is from Carnibacterium. He has managed to pull it out very clearly. So this is how a graph spore staining should be looking. You should be able to observe the malachite green stain in the endospores within the bacterial cell. Okay, the observation should be this clear. This is from 48 hours culture because that's why you are seeing endospores that is being released outside the cell. You can call them exospores, or there are endospores again still present within the cell. Okay. So this image here in right hand side, this is useless. You cannot derive anything out of it. So in 100, always observe in 100x. Doesn't matter it is gram staining, spore staining, or capsule staining. Better to have a individual cells staining, observed. This is an example of capsule staining. Okay, what we are looking is a blue color translucent copper sulfate, induced blue color that is being stained with the capsule present around the bacteria. We are using a negative stain. So between the background and the bacteria, you should be able to see this translucent color. Not like this. Something like this should be avoided. This is the ideal way to take the capsule staining. So once you have studied the morphology, I would say you can also include motility here. Once you are done with anatomy of the cell, you can get into the biochemical characteristics. The first thing usually people do for enterobacteriaceae is Invictus, Intol, August Faustus, methyl red, and citrate utilization test. But many people will argue when you are going for environmental samples, Invic may not be working, Invic is not necessary. But I would say collect as many as information about your bacteria for species diagnosis. Do not constrain yourself by thinking Invic is only for enterobacteria. Why to go for my strain because it is isolated from the agricultural lab, it has been obtained from as an endophytic bacteria from a leaf. No, go for it. Go for Invic. When you are documenting the results of the Invic, make sure you are having an uninoculated control with all the acids. This is the way to document the Invic. This is not the way to document the Invic. Nothing wrong with their results, but they do not have a control here. Okay, without control, do not document. Even though this tube has a control here, why I have suggested not to document it? Better have a white background, basic white background to clear the results, to observe it very clearly. Here it has both a combination of black and white, black color floor with white color background. This will not work. Okay, better have a single background. Then you go for triple sugar ion test where these are the three sugars that is in variable composition. We are looking here what kind of, because of the phenol red, if it is acidic reaction, it will turn yellow. If it is alkaline, it will turn pink. The red color tube that will turn pink, red color because of phenol red. Also, we are observing for gas production. This is an ideal example of gas production. When the gases are produced, your birth region of the plant will be lifted up. Okay. When the gas is hydrogen sulfide, especially, entire bud will become dark black. All right. So this is the inoculation is very much interesting. Usually the buds are made 40% or 30% to the length of the entire tube. The inoculation is by stabbing while pulling it out shrieking. Okay, what we are doing here is we are giving both two kinds of ambient environment for bacterial growth. One is microaerophilic condition, this is aerobic condition. Whatever we are starting, you have done. So we can assess the ability of the bacteria to ferment these three sugars in presence of oxygen and absence of oxygen. Within the same plant, we are doing this. That's why it is very much interesting here. So observe the results. Again, document is take the picture. Go for enzyme screening, whether your bacteria has a catalase or not. Pick a bacteria in a wooden brick. Here you can see the bacterial is there. Dip it inside. This is clearly oxidase, sorry, catalase negative. When you see an effervescence brightly coming out from the bacterial surface, it is catalase positive. Never use a metal stick because some metals they are naturally, they have a catalase activity. So never use a metal stick like this. This should not be done. Also, when you fill a 1%, 3% hydrogen peroxide, make sure it is bubble free. You are going to observe the bubble from the bacterial biomass. If the tube is already having bubble, it will be very difficult to do so. So make sure they are bubble free. I would say 1 ml of 3% hydrogen peroxide can be used for 20 reactions. Okay, maximum of 20 reactions. So 1 ml is very much sufficient. If you are an MSc student or BSc student, 1 ml can be used for the entire class, batch of 20 students. Go for oxidase. These days, oxidase disc is available. So pick up a bacteria, smear on the disc, wait for 15 seconds to observe the disc turning purple, indicating oxidase positive. If it is not, oxidase negative. If any reaction happens after 15 seconds, they are false positive. I would suggest do not consider them. Then we are going for motility check. Obviously, we are studying the morphology of bacteria in, under microscope. We will also study the colony characteristics, also study the broth characteristics. Inoculate the bacteria in the broth, incubate for 24 hours, see how they grow. Some bacteria which likes a microaerophilic condition will create a pellicle-like structure on the surface of the broth to, reduce the diffusion of oxygen. So those, you can see those bacteria that are pakka aerobic, you will see more cloudier biomass near the rim of the broth. Those are microaerophils will settle down at the bottom, or they will be distributed throughout the column. Those are, anaerobic or very much obligatory, facultatively anaerobes. They will settle at the bottom. More biomass will be sedimented. Okay? So after 24 hours, that broth can be simultaneously used for motility test, where pick a drop of bacteria, put it in the glass slide, cavity slide, and observe at the rim of the edges. Because oxygen diffusion is higher here, if the bacteria is motile, all will try to come to the rim. So that's how we will know that they are motile organisms. So do not confuse with the cover slip of the slide along with the rim of the drop. You should be able to distinguish that. So after that, use the same broth culture for antibiotic sensitivity test. You might be wondering why to go for antibiotic sensitivity when you are characterizing a bacteria. Say, for example, if you are working with 10 bacterial species isolated from a soil sample. If you want to distinguish your bacteria, you need to collect as many as data as possible. Okay, say if 10 bacteria looks unique. So I was working with 10 bacteria. So I have inoculated the 10 broad cultures. Now I am smearing the bacteria in the Muller internager. Okay, I'm swabbing them, then placing 5 same antibiotics for the all 10 bacteria in 10 places. Okay. Most of my observation says if the bacteria is of different species, the antibiotic sensitiveness will differ. You may argue that how come it is possible? Antibiotic is sensitivity or resistance is mostly derived from plasmids. But in my observation, I have seen whenever there is a variation in their sensitivity pattern, significant variation, they are usually of different species. So I use this technique to distinguish the bacteria. I don't care about their sensitivity or resistance. So make sure while photographing the ampicillin, amoxicillin, or streptomycin, statin, whatever antibiotic you have used, it should be clearly documented. Okay, the alphabet should be very clear. So whenever you have a ambiguity of zones like this, better go with a larger zone. Measure it. Okay. See here, usually we measure the zone with a scale from the edge to edge, passing through the antibiotics. We will be measuring the diameter. Here, if you should not have placed your disc so close to the rim. If you have done so, better measure the radius and multiply by 2 to get the diameter. Okay. What not to do? Here, the swabbing is not uniform. Here, it is clearly contaminated. There is a, one bacteria is growing as a lawn, another as isolated colony. Clearly contaminated. So these kind of mistakes you should. It's mistake from the handling of the broth. Okay. This just shows how to measure the antibiotic zone. Use the scale. Let the disc circumference to circumference. Let the scale pass through the diameter of the disc. Whenever you have two zones fused or the zone is near, the disc is placed very much closer to the rim, better measure the diameter and multiply by 2. Okay. Always make a table. Okay. With whatever antibiotics you are subjected, make a table. Why? Because when you are done with all this biochemical characterization and antibiotic sensitivity, make a table, make a heat map. Okay. Here, positive means, gram positive means whatever is positive I have given green, whatever is negative I have given red. KOH assay, I used to suggest, or you should do this KOH assay along with Gram's reaction. This is very simple technique. In a slide, take a 3% KOH, 2 drops, and using a toothpick, pick a bacterial biomass and dissolve in this 3% KOH. If the viscosity of the solution increases, it is gram-negative bacteria. If the viscosity does not increase or remains watery, it is gram-positive bacteria. How to confirm the increase in viscosity? Just touch with the wooden stick and pull the solution back. You will see strings coming out of it, which indicates the gram-negative bacteria because they are rich in lipopolysaccharides. All the lipid component has been dissolved in this alkali KOH. That's why you have a viscous solution. So usually I see some people, they do not know when to stop adding ethanol during decolorization processing, gram staining. When your gram-positive bacteria is heavily decolorized, it may appear like pinkish color, but it is light purple, which is also look pinkish. Also, what kind of lamp you use in your microscope? It is a white light or yellow light. Sometimes it is distinguishing between light purple and pink will become very difficult. So I would suggest always complement your Gram's observation using KOH. So here presence of spore is green color, absence is negative, which is red in color. Catalase, oxidase. So what I did basically is for positive I gave green and for negative I gave red. I made a heat map for my isolate. See, I am dealing with 132 bacterial species here. I'm sorry, 32 bacterial species starting from 101. From here, I can clearly, all these 32, I can decide based on this heat map, which are the things that are unique and, what are the things I can consider for sequencing. Because one of my activities also culture collection. I don't want to sequence the same species twice. So I use this heat map along with antibiotic sensitivity assay. Wherever there is a resistance, I have marked it in red. Okay. All these data I will consider. Then I will start grouping my strains based on this heat map. Then I will decide how many unique ones are there. Also, I have a very cheap technique using STS page to identify your uniqueness of the bacteria. I may not know what species it is, but I know what bacteria based on this. Say, for example, if I have a 6 bacillus species, all are endospore positive, gram-positive rods, mostly non-motile. They are all producing amases. So I know they are bacillus basically, but I do not know what is the species. Or I want to distinguish among the 6 strains how many unique species of bacillus is there. So what I do, I will dissolve the bacterial, each and every bacterial biomass in the loading buffer of STS page. I will heat it up and load the sample. What I'm trying to do is to study the profiling of the bacteria, the physiological proteins. I'm trying extracting them and profiling them in STS page. Okay. We all know that during STS page we use BSA, bovine serum albumin, which will usually give one band of 68 kDa. So what I did here is I'm comparing my bacterial strains with this control. Say, for example, I'm giving an numbering system for bacteria, BSA number, I would say. If this bacteria 27, the strain number 27, if it is BSA 004, what it means is they do not have any band which is greater than 68 kDa. That's why it's first zero. They do not have any band which is equal to 68 kDa. That is why it is second zero. And they do not have they have four bands below 68 kDa. That's why 004. So by comparing with, usually gel pictures can be compared visually, or you can give a BSA numbers, and based on BSA numbers, you can also determine the uniqueness of species. See, till here, we know how many species we have. We not know what species it is. We have, based on the characteristics that we have documented, we can compare it with the Burgess manual. We may end up in diagnosing till family or genus level. It is quite possible. If necessary, we can also use selective media to find them. Now, how to identify the species? Go for 6' rRNA. First step is DNA extraction. Extract the DNA, extract the bacterial genomic DNA. Various method is there. Salting out I usually use, or you can go for phenylchloroform. Then go for PCR. Okay. In PCR, what do? You amplify the 6' rRNA gene with the universal primers, 1492 or whatever universal primer you want to use, you can use and amplify it and then go for sequencing. I usually commercially outsource the sequencing. I also do usually, numerical taxonomy. Instead of just producing a heat map with green positives and red negatives, you can also give 1-0 scores for positive one, negative means 0. If you give 1-0 score and sum all the score and divided by number of assays you have used for that particular strain, you will have a tax score or taxonomical score. This score can also be used to find the uniqueness of the bacteria. Once you have this score for all your 32 species, based on similar score, you can again look up the heat map to see whether they are really similar or they vary somewhere else. So tax scores can also be calculated. Okay. So what next? Once you got the sequences, I would say this is a basic idea before getting into lab. Visualize the entire experiment, do it, clean it, get out. This should be the norms in conducting any experiments. These are, very basic things. Here I can show you many of my UG students. They are skillful in identifying fungi, identifying bacteria. Okay, once the sequencing is done, you can use a blast analysis to check with a cutoff score of more than 99% similarity with any sequences, along with your classical data, traditional taxonomical data. You can know what species you are looking at. That's how bacterial species are diagnosed in microbiology labs. I'll be sharing my ResearchGate profile regarding this wrapping experiment that I promised I will share. What are the different kinds of wraps I have used for assessing the growth of the bacteria? That will also be shared along with this. Thank you for joining. Let's know the unknowns.